So now let's talk about horizontal gene transfer. Genes can be transferred from one cell to another cell within the same generation via three processes. Either transformation, where we have one cell taking DNA from another cell as naked DNA within a solution. So this involves a dead cell that is leaking DNA and then a living cell that can take it in. Transduction is the transfer of DNA via a bacteriophage, so a bacterial virus. And then conjugation is transferred directly from one cell to another cell within the same generation using a sex pelus. So looking closer at each of these, in transformation, this was discovered by Frederick Griffith in 1928, and what he noticed was that there were two different types of strep bacteria. Smooth strep that were encapsulated that caused death to the mice that he was infecting. And then they had a capsule around them, and whenever the cells die, or the encapsulated cells got into the bacteria, they caused death or sickness, and then the mice died. But then if he gave the living non-encapsulated cells to the mice, they didn't die. So he thought, okay, so the capsule is what's causing the sickness. So then he thought, okay, I will kill the cells and just give them the capsules and see what happens. Nothing happened. The mice didn't get sick. And so he thought, okay, so the capsules themselves don't do anything. The cells themselves don't do anything. But if the cells and the capsules are together, if you have encapsulated cells, they kill. So I wonder if we just give them the rough cells, the unencapsulated living cells, and then the dead but capsules. And that's what he did. He mixed them in a solution, and then he infected mice. And what he found, the mice got sick and died, and he found living smooth cells at the end. And so this supported the idea that DNA was being transferred from one cell to another cell within a solution. What we know is that cells, to be able to take in DNA, they have to be competent. That means they can take in the DNA. If you have incompetent cells, they can't take in DNA. You can make cells that are incompetent competent by shocking them in a process called electroporation. You basically send electrical pulses through the solution, causing the cell wall to just break apart a little bit, allowing them to be able to take in the naked DNA that has leaked out of the dead cells. And then the DNA gets into the new cells, the cell wall will repair itself, and the bacteria will now have these new characteristics that will be transformed. This video you can look up on Google. I'm not going to look it up right now for you. Transduction then is the transfer of genes from one cell to another cell through the use of a bacteriophage. So there are two types of transduction, generalized and specialized transduction. Specialized transduction is where we're choosing the genes, so we're making the bacteriophage, choosing the genes within that we want the bacteriophage to pick up, and then we can then, or choosing the genes, and then we can infect cells with those specific genes. So that's specialized transduction. Generalized transduction takes any genes. So what happens in generalized transduction is a bacteriophage, infects a virus or a bacterial cell. The viral DNA moves in, cuts up all of the bacterial DNA, uses the bacterial mechanisms, machinery, to produce new viruses, and then viral DNA is pulled in, except sometimes that doesn't happen. Sometimes bacterial DNA is pulled in by accident. And so here you see a virus with bacterial DNA, the purple DNA. That virus, when it infects a new cell, is going to release that purple DNA. The purple DNA does not take over the cell's machinery, but instead gets incorporated into the genome of the new cell. And now he has new characteristics, and all the daughter cells will have that same new characteristics. And then we have conjugation. Conjugation is the transfer of DNA between two bacterial cells by use of a sex pilus. And so the sex pilus is produced by whichever cell has what we call a fertility factor. E. coli are the most well-studied. E. coli are, I think, the favorite bacteria of most microbiologists. So when you have two cells, you have two opposite mating types. You have the F plus and the F minus cell. F plus cell has a fertility factor plasmid that might contain other characteristics too, like resistance or the ability to use certain nutrients. The F plus cell will produce a sex pilus that will attach the two cells together. The plasmid will replicate and then will move from the F plus cell to the F minus cell. And then the cells will release, and now you have two F plus cells. And here's bacterial conjugation. So that was chapter 8 in a nutshell. And now we're going into chapter 9, which is the biotechnology chapter. So we've talked about microbial genetics. We've talked about mutation rates, and now we're going to talk about why we use microbes in biotechnology. What microbiology has to do with biotechnology. So in this chapter, we'll look at how biologists use biotechnology to amplify DNA so that we can then isolate new genes or determine paternity or determine if there's a possibility of a mutation occurring within a cell. And then, oh, I told you this maternity right there, or paternity for that one. So biotechnology, in a nutshell, is the use of microbiological or biochemical techniques to solve problems. So we have a problem, we need to use some way to solve it. An example, maybe we are trying to genetically alter an organism so that it can grow in the desert where it normally can't grow. Or maybe we want to produce bigger, healthier, tastier tomatoes. Or maybe we just want to make glow-in-the-dark fish because, hey, who doesn't like glow-in-the-dark anything, right? The first thing we have to do is we have to be able to mess with or to get the information from our DNA that we care about. And to do that, we have to use DNA scissors or restriction enzymes. Restriction enzymes are enzymes produced by bacterial cells for the specific purpose of protecting them from bacteriophages. So they're basically the immune system of a bacterial cell. They cut all DNA, any DNA, it does not matter, at specific regions that they recognize. The regions are typically palindromic, which means they read forward and backward the same way. So MOM, M-O-M, is a palindrome. Here's a palindrome, G-A-A-T-T-C. Down here, G-A-A-T-T-C. That's a palindrome. It reads forward and backwards the same way. Restriction enzymes can produce what we call sticky ends. So this is a sticky end where you have a region of the DNA that hangs out that doesn't have a complement. And with this, we can actually hybridize new DNA to it. Or it can produce blunt ends where it cuts the DNA into two complete sections. So here ALU1 produces a blunt end, and it cuts DNA between AGCT. It cuts between the G and the C every time, so it produces a blunt end. Here's BAMH1, cuts between the GG in a sequence of GGATCC. GGATCC. So it cuts between this G, and it just moves across. And so it produces a sticky end. So once we can cut DNA, and we can then, if we use the same restriction enzymes and cut other DNA, we can hybridize them together, and then we can insert the material into a cell, and we can clone the material. So this is how we can produce a lot of DNA, or we can produce proteins, or whatever else we might want. So here's an example of how this might work. You take a bacterial cell that has a plasmid. You take the plasmid out. The plasmid acts as a vector. You take DNA that you care about from a genome. So here's the gene we care about. We cut it using a restriction enzyme. We cut the plasmid using the exact same restriction enzyme. We then insert the gene that we care about into the plasmid, and we insert the plasmid into a cell. We then allow the cell to replicate, and it might produce lots of proteins. In this case, we're producing cells that can clean up oil spills, or maybe cells that contain genes for pest resistance that we can insert into plants. Over here, we're producing proteins, either for medicine, so human growth hormone for someone who has a very shunted growth, or maybe we're producing proteins to produce clothing. No matter what, we're going to have to isolate the DNA that we care about and cut it with the restriction enzymes and insert it into a plasmid. Now, all of this occurs on that, again, microscopic level. We don't actually see this. We insert this into a cell that is competent, and for E. coli are not competent naturally, so we use electroporation to shock them to make them competent. And so there's electroporation that you can see. So this is all occurring within a solution, and so we're not actually seeing this happen, which means we have to be able to select those organisms that do have characteristics we care about. Sometimes the vector that we choose can be called a high copy number vector, and those vectors are going to replicate much more rapidly than even the cell. And so each cell will have multiple copies, and as the cells replicate, more and more copies of the vector will replicate as well. So a good vector is going to have more than one selectable marker. You're going to have at least one selectable marker to allow it to grow where other cells would not grow, and then another selectable marker that allows you to choose those cells from other cells. So you want it to be able to grow, so it has to be selective, but it also has to be able to be differential. And a good vector is going to be both selective and differential, kind of like MSA media. So here's an example. A marker that has a resistance factor and has the ability to produce an enzyme that breaks down lactose, so the LAC gene. And so if you're in the presence of a lactose media, the LAC gene will break down the lactose, forming a blue blue color. But if you insert the gene that you care about into the plasmid, it breaks up that LAC-Z gene, kind of like that transposon. And then the LAC-Z gene becomes non-functional. So then when you grow the bacteria on medium containing both the whatever the antibiotic is that vector contains resistance to, and lactose, you're going to produce both blue and white cells. The white cells are the ones that can't use the lactose, so they don't produce that blue color. And that means that they have the gene that you care about inserted in that LAC-Z gene. So here's my example of that exact same thing, okay? Here we have a plasmid. The plasmid has antibiotic resistance, and it has this area here, this red, which is a gene that codes for beta-galactosidase, which also breaks down lactose. Here's a sequence for insulin, okay? So we're going to cut both the plasmid and the DNA with the same restriction enzymes, and then we allow them to splice together or come together. So you're going to have two different types of plasmids at this point. You're going to have a plasmid that contains the gene of interest that's inserted in between, so it breaks up that beta-galactosidase gene. And then you're going to have some plasmids that just come back together. They hybridize back together. So there's no gene in there. And you're going to take bacteria, specifically E. coli bacteria. So think of all of these are E. coli bacterial cells. We add them to the solution with these plasmids. We can't see any of this. And what we're going to find are that some E. coli are going to take in the plasmids with the gene of interest inserted. Some bacteria are going to take in just the plasmid without the gene of interest, and some will take in nothing. So they did not maybe become competent, or maybe they just, their cell walls fixed and they never just took in anything. So you got these three different types of cells that are replicating. You got millions of each type, right? In the solution. But again, they look the same. So if we plate them on a basic nutrient media, all colonies will look identical, and you don't know which one is the one you care about. If we plate them on a medium containing that antibiotic resistance, so in this case ampicillin, then only those cells that have ampicillin resistance are going to grow. And so now we have only, and I'm using just one colony for each, but you get the idea, only the ones that have the plasmid. But if we plate them on a media containing ampicillin and lactose, now only the cells that contain the gene of interest are going to turn white because they can't break down the lactose, while the other cells will. And so now we can take these guys off the media, direct selection, and we can grow them. All right, I'm going to stop this video and we'll get into the next material. Okay?